Hello everyone, this is my first video series on my review crash course on physiology. So the topic that I will be discussing will be cell physiology part 1. For the outline, principles of cell and the membrane function, the cell membrane, the membrane transport. So this is an example of the cell membrane or the plasma membrane. So the cell membrane has a 5 nanometer thick lipid bilayer with associated proteins. So it is composed primarily of phospholipids, which is 25%, and proteins, 55%. For its functions, selective transport of molecules into and out of the cell, cell recognition through the use of cell surface antigens, tissue organization, membrane-independent enzymatic activity, determination of cell shape by linkage of the cytoskeleton to the plasma membrane. It is made of lipid bilayer, also known as the fluid mosaic model. The outer leaflet, composed of phosphatidylcholine, sphingomyelin, and phosphatidylethanolamine. For the inner leaflet, it is composed of phosphatidylserine and phosphatidylinositol. Membrane lipids. The major lipids of the plasma membrane are the phospholipids and the phosphoglycerides. A minor lipid component of the plasma membrane or the cell membrane is the glycolipids. These lipids, as their name indicates, consist of two fatty acyl chains linked to the polar head groups that consist of the carbohydrates. The glycosyl phosphatidylinositol or the GPI is an example of a glycolipid. It plays an important role in anchoring the proteins to the outer leaflet of the membrane. Cholesterol is a sterol molecule, is also a critical component of the bilayer. It is found in both leaflets and serves to stabilize the membrane at a normal body temperature. Both cholesterol and glycolipids, like the phospholipids, are amphipathic. They are oriented with their polar groups on the outer surface of the leaflet in which they are located. Their hydrophobic portion is thus located within the inferior of the bilayer. Phospholipids are amphipathic molecules that contain a charged hydrophilic head and a two hydrophobic fatty acyl chains. The hydrophobic fatty acyl chains form the core of the bilayer, and the polar head groups are exposed on the surface. For the membrane proteins, it is the 55% of the cell membrane. These membrane proteins are classified as the integral, lipid-anchored, or the peripheral. The integral membrane proteins are embedded in the lipid bilayer, where the hydrophobic amino acid residues are associated with the hydrophobic fatty acyl chains of the membrane lipids. Glycosyl phosphatidylinositol or the GPI anchors proteins to the outer leaflet of the membrane. Peripheral proteins may be associated with the polar head groups of the membrane lipids, but they are more commonly bind to the integral or the lipid-anchored proteins. Another cellular function of the cell membrane is the membrane transport. The transporters have been classified in several different ways. It is divided into 4 general groups. Number 1 is the water channel or the aquaporins. All isoforms allow the passive movement of water across the membrane. Some isoforms provide a pathway for other molecules such as glycerol, urea, mannitol, purines, pyrimidine, carbon dioxide, and ammonium to cross the membrane. At the cellular level, each aquaporin molecule consists of 6 membrane-spanning domains and a central water-transporting pore. 4 aquaporin monomers assemble to form a homotetramer in the plasma membrane, with each monomer functioning as a water channel. An example of this are the cells in the collecting ducts of the kidneys. Aquaporin 3 and the aquaporin 4 found in the basolateral membrane, and the aquaporin 2 on the apical membrane. Moreover, the abundance of aquaporin 2 in the apical membrane is regulated by the antidiuretic hormone, also called arginine vasopressin, which is the crucial for the ability of the kidneys to concentrate the urine. Another transporter group is the ion channels, important for the function of the excitable cells. Example of these are the neurons and the muscle cells. It is classified by their selectivity, conductance, and the mechanisms of the channel gating, which is the opening and the closing of the channels. Selectivity, only a specific ion through. Channel conductance, a number of ions that pass through the channel. Channel gating, ion channels fluctuate between the open state or the closed state. The third transporter group are the solute carriers. These carriers can be divided into three groups according to their mode of transport. The uniporters or the facilitated transporters transport a single molecule across the membrane. The transporter that brings glucose into the cell. Glucose transporter 1, GLUT1, or SLC2A1, is an important member of this group. The symporters or the contrasporters are the second group that couples the movement of two or more molecules or ions across the membrane. They also transported in the same direction. An example of this is the sodium-potassium chloride symporter found in the kidneys, which is crucial for diluting and concentrating the urine. The third group, the antiporters or the exchange transporters, also couples the movement of two or more molecules or ions across the membrane. In this case, however, the molecules and ions are transported in the opposite direction. An example of this is sodium hydrogen antiporter or NHE1. The fourth membrane transporter group, which is the adenosine triphosphate dependent transporter, is divided into two groups. The first group is the ATPase ion transporter, which is subdivided into two groups. The P-type ATPases are phosphorylated during the transport cycle. Sodium-potassium ATPase is an important example. It plays a critical role in establishing the cellular ion and the electrical gradients as well maintaining the cell volume. Second subtype is the V-type ATPases, found in the membranes of the several intracellular organelles, such as the endosomes, lysosomes. As a result, they also referred to as the vacuolar H-ATPases. It plays an important role in urinary acidification. The next group is the ATP binding cascade or ABC transporters, which is a large group of membrane transporters, amino acid domains that bind ATP like ABC domains. It transports a diverse group of molecules or ions including cholesterol and chloride, bile acid, drugs, iron, and organic anions. To be continued. Thank you for watching.